In this video, we will discuss about the Siglec-10 expression on pancreatic cancer cell line. So in the previous video, we have understood the Siglec-10s are present in the tumor microenvironment of the pancreatic cancer, and that we have validated using the publicly available data, and also in this TGCA, which is the cancer genome atlas, So now they wanted to check whether Siglec-10s are also expressed on the pancreatic cancer cell lines or not. And for control they use the ovarian cancer cells, and since it is well identified that Siglec-10s are expressed on the ovarian cancer cells, so they use this ovarian cancer cells as a control, and the antibodies that they used as a control is CD24, and CD24 is recently been identified as a glycoprotein ligand against this Siglec-10. So what it is, they took these different pancreatic cancer cell lines such as ASPC1, MIA, PACA2, PANC1. These are the three pancreatic cancer cell lines they took, and then stained them with CD24, and also stained them with this recombinant, or we can say that chimeric Siglec-Fc, or Siglec-10 chimeric-Fc. Yeah, this is the right term, Siglec-10 chimeric-Fc, that can bind to the Siglec-10. And we can see that for the case of ASPC, both the expressions are there, but however the expressions of this Siglec-10 Fcs are higher, where the expressions for the CD24 are lower. And in MIA-P PACA2, the expressions are higher for this CD24, but lower for this for this Siglec-10 expressions. And similarly, they have seen the expressions for the PANC1 follows the same trend, that this seems to be higher. These are higher for the Siglec-10 Fc, signals are higher for Siglec-10 chimeric-Fc, but slightly lower for CD24. And here also I said it the opposite thing, that it is higher for Siglec-10 Fc and slightly lower for anti-CD24. And these things they have quantified on the panel B, D and F respectively, where we can see that signals for the Siglec-10 ligand cells are significantly higher for ASPC1, and they are also higher for MIA, PACA2 and PANC1. And for both the cells that are expressed this CD24 and Siglec-10 are lower compared to the case where Siglec-10s are only expressed, or Siglec-10s or CD24s are expressed, as this should be evident that both of them, some of them will express only one ligand because of the difference in the expressions of the Siglec-10 and CD24. And the overall expressions are shown here. Here also we can see that the expressions for the Siglec-10s are higher compared to the expressions for the CD24. And when they check it for the ovarian cancer cells, they see a different thing, that is the expressions levels of CD24 and Siglec-10s are almost similar in all the three cases, that is ovarian SCOV3, ovarian cancer 8, OVCAR 8 or MCF7. And these things they have quantified in the panel H, J, L, where we can see that the expression levels, in some cases even the expression levels are high for CD24 for, you can say, all the three cases. So it is, the results are like reverse that they have seen in the pancreatic cancer cell. However, the common things that they have seen, that both the cells has Siglec-10 expressions, that is verified by CD24 and Siglec-10 chimeric-Fc protein. Here the total levels are almost similar in all the three cases. And for control, they to ensure that the Siglec-10 chimeric-Fcs bind to the Siglec-10s and it is sialic acid dependent, what it is, they mutated this R119 with A, which abrogates the sialic acid binding capabilities. And in that case, they are able to see reductions in this Siglec-10 Fc proteins, Siglec-10 Fc signals that can be seen in all the cases. So this indicates that the Siglec-10s are present and it is sialic acid. The signals are the Siglec-10 chimeric Fc proteins is sialic acid dependent. So that's all. They have concluded that Siglec-10 expressions are present on the pancreatic cancer cells, and they are the subset of CD24 cells. So that's all for this video, and if you need to understand more about this results or this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.